Good morning, all. Today topic is protoplast culture. Yesterday we completed tissue culture, totipotency, techniques, steps in tissue culture or totipotency, and applications of tissue culture, applications of totipotency. These are all what discussed yesterday. And today topic is protoplast culture. First of all, what is the protoplast? Where it is? If you take the protoplast, this is the plant cell. So next to green color plasma membrane. Plasma membrane. Inside the plasma membrane, nucleus present, and around the nucleus, remaining cell organelles are present. CO means cell organelles, such as mitochondria, Golgi complex, ribosome, so on. These are all. So this is the cell. If you separate this is cell wall. The red color part is cell wall. This is the wall. This is the wall. This is cell wall. And except the cell wall, next layer is protoplasm. Sorry, next layer is plasma membrane. Now this entire portion from plasma membrane, this is plasma membrane. The only green color plasma membrane. This is nucleus. And between the plasma membrane and nucleus, cell organelle present. That's why this cell organelle content pores called as cytoplasm. This is called cytoplasm. This is called cytoplasm. Cytoplasm. So this is cytoplasm. In middle nucleus. Now both called this nucleus cytoplasm. This both together called as protoplasm. Now this both. Let's change color. Let same yellow color. If you take only yellow color border, that is cytoplasm. Including nucleus, that is protoplasm. With the plasma membrane, that is protoplast. With plasma membrane, that is called protoplast. Now this entire portion, now this is complete cell. In cell, I have the cell wall and green color plasma membrane. Inside the plasma membrane, protoplasm there. What is protoplasm? Nucleus and all cell organelles. If you separate the nucleus, the rest of the portion called as cytoplasm. That's it, simple. Now the simple, we want only what is protoplast. How it is culture the protoplast. For this, the protoplast is in cell, except the cell wall, except the cell wall. This red color, except this red color portion, except this red color one. Rest of the all called as, rest of the one called as protoplast. Now this entire structure called as protoplast. Now this is the act as an explant. Now the explant is protoplast. So far we know the explant is any part of the plant, maybe cell or tissue or organs. So they are act as a explant. But in this, the explant is protoplast. The protoplast separated from the cell, right? So simple definition, the plant cell without the cell wall, clearly given plant cell without the cell wall called the protoplast. But they possess inside the protoplast, possess plasma membrane. Diagrammatically, you can see plasma membrane, all other cell components. Protoplast of different species can be fused to generate hybrids. This process referred as somatic hybridization. That means the simple somatic hybridization means fusion of protoplast. One protoplast different different from different species. Protoplast fuse with another protoplast, different species. So as a result, formation of hybrid form. This process referred to as somatic hybridization. And hybridization is the phenomenon fusion of normal protoplast with a nucleotide protoplast that result of formation is hybrid. I will explain later hybrid. First of all, let's discuss about somatic hybridization. Somatic hybridization or protoplast culture. Don't get confusing this word. In examination, they can ask write about protoplast culture or somatic hybridization. Describe somatic hybridization. Both are same. Hybridization, hybrid different. Of course, similar. Both are similar, but the small changes between the somatic hybridization and hybridization. Simple somatic hybridization, a fusion of both protoplast, but in hybridization, a fusion with the cytoplasm. Protoplast fuse with cytoplasm. That's it. That means in one, for example, in somatic hybridization, this is protoplast. When it is called protoplast with nucleus present, that is called protoplast. Plus, so another nucleus. So this is called somatic hybridization. This is called somatic hybridization. But what is hybridization? In hybridization, uh, one is common. This is common. That means this one as it is protoplast. But another one is only cytoplasm. Only cytoplasm. No nucleus. So that is called hybridization. Right? Now let's see. Right. Clear. So what is protoplast culture or somatic hybridization? Clear. Right? Now you know where the protoplast. Now you have to separate the protoplast from the cell. Where is protoplast? Inside the cell. So inside the cell protoplast, you have to separate it. That is separation by two methods. One is mechanical method. Mechanically we can separate it, or enzymatic method. First, mechanical method. In mechanical method, we can use large, highly vacuated cell storage tissue. These cells are plasmalized in isosmotic solution. Finally, the tissue dissected. Simple, what happened here? For example, this is the cell, isolated cell. To tend to be isolate from the cell. First of all, these one placed in. This is the solution. This solution have the high concentration when compared with the cell concentration. What happened when this cell, so first of all in common, the cell covered by cell wall. In isolation separation, first the cell wall present, okay. You have to separate the cell wall, right? For this, in mechanical method, first storage inside the high concentration solution. What happened if the cell placed in high concentration solution? This is high concentrated solution and cell nucleus, cell wall. Actually, this is full-fledged cell. That is the cell content completely filled with. Now, when this cell placed in high concentration, when you place it in high concentration, what happened? You remember, always water move low concentration to high concentration. Water move from low concentration to high concentration, low to high concentration, low to high concentration. Now tell me where the low concentration, already told you low concentration in cell, where high concentration in the beaker solution. Due to this reason, from the cell, cytoplasm water uh, goes out, comes out. As a result, what happened? This full-fledged cell, actually, we cannot separate the cell wall and plasma. They are both firmly attached in full-fledged cell. In fully fledged cell, what happened? The plasma membrane firmly attached to the cell wall. But when actually this is full attached to the cell wall because of full content of water. But what happens when the cell is placed in high concentration solution, automatically the cell content comes out. That's why the pressure decrease. As a result, completely the plasma membrane separated from the cell wall. Like this appears as a now finally it is appear as a fine first cell wall as it is common, cell wall common as plasmolysis. This process called plasmolysis. Plasmolysis. Due to the plasmolysis, what happen here? Cells shrink. Cells shrink and common as it is. Now the gap is formed. Now the cell wall separated. Now from this cell wall separate. Now from this we can separate from the cell wall. Cut the cell wall. Now only protoplast remain. Now we separated this solution. So this is the mechanical method. And another mechanical method also with the help of the scissors. And finally, what happened? After plasmolysis, we placed it in the microscope, special type of microscope, and cut with scissor. Cut the cell wall with the help of the scissors. Next one, enzymatic method. In enzymatic method, we can use cellulase enzyme and pectinase enzyme. Why you use cellulase and pectinase enzyme? Do you know the cell wall structure, cell wall chemical? In cell wall chemical, mainly made up of magnesium and pectate, calcium, magnesium pectate. And also the cell wall made up of cellulose. For example, if you take the cell, this is the middle lamella, either side primary cell wall. And this is one cell. This is the another cell. If you take tissue, in tissue group of cell, I have selected two cells only two cells. Actually, this is the group of cells. Like this, the cell each attached one above another, one cell to another cell, one cell to another, finally formed as a tissue. Finally formed as a tissue. But in enzymatic method, what happened? These enzymes, with the help of this enzyme, the cell wall degrades. Cell wall degrades. Already told you the primary wall layer by cellulose. In middle lamella, magnesium, calcium pectate. So if you use pectinase enzyme, this pectinase enzyme dissolve the middle lamella. As a result, the both cells are separated and cellulase enzyme which act on the primary cell wall cellulose. Now the cellulose dissolve. Now what is remain after cellulose dissolve? Only remain the protoplast. Only remain the protoplast. Now the protoplast is separated out. So this is the isolation of protoplast. This is the isolation of protoplast, mechanical method and enzymatic method. Right? And next one, you can see here in enzymatic method what happened. After plasmolysis cell cut with the scissor. Now the plasmolysis, this protoplast separated. Later the fusion of protoplast. Right. So anyway, after separated this protoplast method, actually in this both actually, first of all, this mechanical method somewhat the complex to separate the protoplast. Enzymatic method easy method because when you use the enzyme, they easily protoplast are separated, right? And methods of protoplast culture, uh, liquid cultures, agar culture, droplet culture, co-culture, and root culture. Sorry, no, no, right. First, liquid culture. In liquid culture, the early developmental stages of protoplast, because it allow easy dilution of transfer, protoplast easily get divided in liquid medium. That is the first one. Next, agar culture. So, in liquid culture, we culturing, that means the substrate is the liquid manner. Why it is used the liquids? Because it dilution easy, transfer easy, so can grow easily. That's why use a liquid culture medium. And agar culture, most frequently used solidification protoplast culture media. Protoplast suspension is taken a double density and mixed with melted agar medium at 45 degrees centigrade and mixed well and plated in the petri dish. Simple. Already in this petri dish, already taken a agar and the agar embedded the protoplast. Simple. And agar embed, this is the protoplast. This is agar. This is petri dish. Right. Droplet culture. In this culture, suspending protoplast in liquid culture, here also liquid culture medium placed on the petri dish. Simple. In the form of droplets. In the culture, protoplast clumped together at the center of droplets. Simple. What happened here? In suspension protoplast, then liquid culture, you have to take the not a solid like agar, petri dish, liquid culture media. This is liquid culture media. And from the droplets, you add protoplast, droplets like drops, like drops, droplet, the culture media use. Co-culture. Next, protoplast of two different plant species, one slow growing and another fast growing, can be co-cultured. In this, you have to take the two different plant species. Protoplast of, you separated the protoplast by any method, mechanical or enzymatic method, two different plant species. In these two different plant species, one is slow, one is fast. Now this is co-cultured. This type of culture advantages since growing species provide the growth factor and other chemical which help generation of cell wall and cell division. That means actually one is slow, one is fast, but later next offspring is grow fastly. And another handling droplet culture and immemorialization bed layer. So they are the another technique of the protoplast culture. And to take hybrid hybridization, hybrid, where it is right. Planting protoplast, fusion of protoplast, somatic hybridization completed. And another one actually, one is forget in protoplast culture methods, chemical fusion and electric fusion, chemical fusion and another one electric fusion, right? Actual hybrid not given, I think here. Hybrid given or not? Fusion of protoplast, somatic hybridization, okay. Types, okay, clear. Culture of the protoplast, clear. Variability protoplast culture, right? Mechanical completed, enzymatic completed, Culture of protoplast, okay. Fusion of protosomatic hybridization, fusion. So liquid culture, they are completed, but not completed chemical fusion, electrical fusion. Complementary section because section trading. Regeneration of hybrids, other methods. Right, hybrids also given. I will give later. Before that, chemical fusion. In chemical fusion, employing polyethylene glycol, polyethylene polyethylene glycol, PEG, polyethylene glycol, which is first developed by Coho and Michelak. About 0.6 PEG solution, this PEG solution dissolved in 1 gram of glucose, CaCl2 and 0.7 Kh2PO4, added a drop of pellet protoplast culture tube. Already this is protoplast culture tube, protoplast culture tube. Already inside of the separated protoplast. For example, this is the protoplast. In this protoplast, we added the chemical, PEG chemical added, polyethylene glycol, PEG chemical added, and also added glucose, KH2 PO4, KH2 PO4 added drop pellets. After having capped the tube, next after that, PEG chemical use and glucose added. Next CaCl, sorry, KH2SO4 and also CaCl2. These chemicals are all added in this tube. Later, closed by cap. Protoplast PEG are incubated at room temperature for 40 minutes. Occasionally, rocking of tubes helps the brings. Just shake the tubes, which helps the bring the protoplast in contact. This followed by elution of PEG by addition 0.5 ml of protoplast culture. The medium of the tube after every 10 minutes. Protoplast has first brought close together. What happened already? You have taken the two different protoplast here, A protoplast and B protoplast, both protoplast. After that, you added the PEG chemical CaCl2, KH2SO4, they are all. But these are triggers to fusion both the protoplast. Both the protoplast. What happened? If the both are close together, aggregated, what happened finally? The PEG dilution due to the disturbance created in plasma membrane. Now finally what happened? Both protoplasts fused, then transferred into the culture medium and induced the callus. When after the fusion due to the chemicals, now these are transferred into the media, and culture it. Culture process same method, same step what we discussed yesterday, the same, cultured into media. So this is the chemical fusion. And another one is electrical fusion. So in electrical fusion, what happened? The cells, the whose cell walls were removed, undergo conversion of shape from the cylinder into shape. How the, you know already how the cell wall removed. After cell wall removed, the protoplast only remain the protoplast. Later, in this method of fusion of development, right, in this method of fusion of developed by Senda scientist in 1979, one droplet containing mixture of each parental protoplast is placed between two microelectrodes. In this, instead of chemical, we use microelectrodes, and with help of the micro manipulator subjected to the difference potential of about 10 volts to 15 volts. Then, due to the volts impulse, protoplasts then aggregate into long chain, which is perpendicular to the electrodes. The fusion is induced 1 minute later by brief high electrical pulse, that is mostly 100 to 150 volts for 10 to 20 microseconds. The high voltage DC pulse induces the reversible breakdown of the plasma membrane. In first method, chemical method, PEG dissolve the protoplast, but here the the current impulse, DC pulse, induce the reversible breakdown of the plasma membrane at the site of the cell contact. Now it is leading to the fusion consequent member of reorganization. The entire operation is carried out manually in a special designated equipment called electroporator. This is done not in tissue culture tubes, not in cultural tube. It is only done in the special type of operator designated equipment that is called electroporator. And while the functioning happened, we have to observe the under microscope. The fused protoplasts are then transferred onto the culture media. Simple. After fusion, then transfer into the culture media. The rest of the process same. After transfer into culture media, sterilization, incubation, inoculation, incubation. So they are the common characters, right? So this is electrical impulsion. So this is the protoplast culture, and next one, hybrids. Hybrid mean hybrids are cytoplasmic hybrids are produced in variable frequencies in normal protoplast fusion. Experiment and contain the nucleus one species but cytoplasm from the both plant parents. They are formed due to the elimination of the nucleus of one species from the normal heterokaryon. Already told you that is the fusion occurs between protoplast with nucleus, another only protoplasm, no nucleus, without nucleus. Now that is called hybrid. After hybrid formation, now it transfer into the culture media. Simple. The transfer cytoplasmic male sterility from Nicotiana tobacanum, Nicotiana silvestris. These are the examples. So what happened in this? Mainly the hybrid approach has been used for transfer of cytoplasmic male sterility. What is cytoplasmic male sterility? Cytoplasmic male sterility means in male reproductive organ pollen grain of tobacco. For example, this is the tobacco pollen grain. Actually, tobacco pollen grains also in each cell the nucleus present and cytoplasm and cell wall present. But in what happened in tobacco, the inactive. Nucleus is inactive and it is shows the sterility. So that's why this is can use as a hybrid formation. And the importance, what is the importance of this protoplast culture? The main achievement of somatic hybridization. The best one already the sexual crosses in plant breeding program often failed produced hybrid due to the incompatible barriers due to any reason. For example, there are some plants are there. There might be the reasons are more to do not fertilization. So because one of the sexually incompatible two, they are mainly pre-zygotic embryo. Even actually first they did not form embryo. But if even embryo formed, that is premature. So these are the barriers. These barriers in sexual hybridization have been overcome by somatic cell fusion, protoplast culture, somatic hybridization, and hybridization. A few somatic hybrids between two incompatible species have found application in industry and agriculture. Scheder obtained amphi-diploid Datura inoxia, Datura discolor, and Datura inoxia, Datura stomanium by fusing their diploid mesophyll protoplast. Actually, vegetative cell diploid. Vegetative cell in what happened? Datura, the vegetative cell, Datura, mesophyll cell diploid. And here also another mesophyll cell diploid. Now the both diploid cell, vegetative cell, protoplast separated by mechanical or enzymatic or any method. After that fusion takes place. As a result, what happen? It is from polyploid condition, foreign condition, from the foreign condition. So by fusing their diploid mesophyll protoplast, these interspecific hybrids are now propagated for industrial uses as they produce higher, for example, scopolamine content than parental forms that is present in Datura. For that purpose, we can produce more number of Datura plants. Intergenetic hybrids have been reported in somatic hybridization between tomato and potato. This somatic hybridization not only in the same species, this somatic hybridization can perform between the two different tomato and potato, and also Brassica napus and Thalapsi piriforms with high contents of nervonic acid. Uh, so, and another one is given, okay, two enough. One is Datura, another one is potato and tomato. So, between the crosses, potato protoplasm and tomato protoplasm. So, anyway, the simple one word, the mainly somatic hybridization where the plants are failed to sexual reproduction, they fail to form gametes, or fail to form production of plants, then only we motivate towards the somatic hybridization. So this is the advantages of somatic hybridization and its hybrids. The next one, let's see some of the important topic in molecular biology. In biotechnology part, in biotechnology, one block that is in biotechnology tissue culture. Don't get confused with the tissue culture and totipotency. Both are same. Tissue culture process, totipotency. Based on the totipotency property, tissue culturing occurs. That is the ability of division of cells. That is called tissue culture. And application completed. Any procedure. Anther, pollen, and ovule culture. 14.8. Here the explain different. Anther different, pollen different, ovule. But anther different, but process is same. Process. The steps are same. Don't get confused those steps. Right. And next one. Fourth back, biotechnology, transgenic plants. I will explain later about transgenic plants. This is also important. But before that, now the one remaining part is there in molecular biology. That is the most important topic and very interesting topic. That is regulation. That is page number. Gene expression, sorry, regulation of gene expression in eukaryotes. Sixth in it. Regulation, eukaryotic translation. Eukaryotic transcription. Eukaryotic transcription completed. Translation completed. Now regulation. Your regulation of gene expression in eukaryotes. Right. White screen అది. Just wait, let me take board. Okay. Right. So what is gene regulation first? Gene regulation, that means stop, switch on, switch off of the transcription translation. You know the central dogma. In central dogma state that the genetic material, genetic material, DNA form mRNA, on the mRNA stand protein, protein form. This is central dogma. So these proteins are enzymes. The enzyme express the characters, act on substrate and express the character. So this mechanism regulated. Let's see how this mechanism regulated in eukaryotes. In bacteria, prokaryote, the gene regulated maintains an internal flexibility. Only it is occurs internal, turning genes on and off in response to environment changes. There is no factors. In response to environment changes, the genes turn on and off happen. But in multicellular eukaryotic organism, the regulation brings about cellular differentiation. Let's see some of the different type of regulation gene expressed in eukaryote. First, Britton and Davidson model. In Britton Davidson model, in gene structure, we take the gene, some portion, this is producer gene, produce, producer gene part. Here, operate, over operate. Here, regulated gene there. Sensor site. And this is promoter. This is actually promoter. And after recognition site or promoter, when the polymerase enzyme promote here, now this operated by operator genes. Now it can move forward. On the producer genes, it forms the RNA. Forms the RNA. If any changes occur in this, switch on, switch off. If it is activity, switch on. It is inactivity, switch off. When switch on, RNA formed. After RNA formed, protein also formed. But in switch off, automatically does not form any RNA and does not form protein. And this is the one method. And second one is chromatin structure and control of expression, gene expression. In the second method, the chromatin structure also can control gene expression. Let's see how the chromatin structure controls gene expression. First of all, chromatin. Do you know chromatin structure? Chromatin, DNA plus histone protein, H protein. DNA plus histone protein. DNA plus histone protein. Actually, these two together called as a nucleosome model or solenoid model. As a result, such elongated chromatin thread compactly arranged. Actually, this DNA compactly arranged with the H protein within a small nucleus within a small cell. This is also limits to access regulator factor to their target site. This leads to chromatin induced depression. So that means, for example, this is DNA, chromatin structure, histone protein, and again DNA, again here histone protein, DNA. For example, where the RNA partner is present, it's moved to forwards to synthesize RNA. When reach the histone protein, can it move forward? It cannot move forward. When reach the histone protein, it cannot move forward. Stop here, it would be stopped. That's why it is act as a repression. The example here, SWI SNAT protein. When this protein, when this protein present in cell nucleus, it is important to formation of tightly bonded histone protein. Sorry, induced repression. Sorry, this leads to chromatin induced repression. When you apply the Swiss SNT protein complex, this protein complex remodeling the chromatin nucleosome model, right? Remodeling means again form histone protein, again form histone protein. That's why this protein stops the regulation process, stop the uh, transcription translation process. And where they mainly present these, that means this type of packing, the histone protein tightly packed present mainly heterochromatin. If you take the chromatin, there are two types: heterochromatin, euchromatin. Heterochromatin, euchromatin. Heterochromatin, it is more densely packed. Heterochromatin more densely packed. Here, dark color. Here the loosely packed, light color. This is transcriptionally silent because if it is tightly packed, they cannot move the polymerase enzyme, cannot form the RNA. That's why it is silent. But here somewhere we can easily separate these stones in euchromatin portion. That's why it's transcriptionally active. If any changes occur in chromatin alteration in gene expression, methylation takes place or acetylation also takes place. What is methylation? I will explain about the methylation and acetylation in the epigenome. Right? The next one, what is given in next one? So again, actually this epigenetic outside, but here the this methylation inside. Let's see the methylation topic here. The DNA methylation is a biological process by which methyl group added to the 5' DNA strand cytosine. For example, this is the DNA 5' 5' 3' 5' DNA strand cytosine. For example, here the cytosine present, cytosine in DNA. Already guanosine cap present mRNA, but in DNA, it is DNA here cytosine present because guanine opposite cytosine present. In cytosine, in cytosine attached by methyl group, attached by methyl group. When it is attached by methyl group, this process called as methylation. This DNA methylation regulate gene expression by recruiting protein involved in gene repression or by inhibitory protein binding receptor site. When CH3 group attached to the cytosine, automatically here the promoter site or recognition site, this recognition site attached by repressor protein, RP, repressor protein. When it is occupied by repressor protein, there is no space to bind the polymerase enzyme. Without polymerase enzyme to bind the specific site, how can move the polymerase enzyme? How can synthesize RNA? And how can form the protein strand? That's why when the methyl group methylation takes place, automatically the rest of the following process are retarded. So this is methylation. And then what trigger DNA methylation? And DNA methylation mainly forms in the course of life, aging process, environmental changes, the lifestyle factor such as smoking, diet, induced biochemical alteration, DNA. So that means what happened? For example, I can let me take the one example. Um, can you work the same capability with 30 years age and with the 60 years age? We can't do because when reach the 60 years, when 60-year-old, automatically some of the cells methylation takes place. Due to the methylation, there is no formation of protein. There is no formation of protein. There is no energy. There is no body formation. That's why automatically, and all some of the characters loss. For if you take the digestion process also, for example, digestion enzymes. These digestion enzymes are proteins. These are digestion enzymes, transcript translation formed by transcription process, translation process. This is normally occurs in years and years. When these enzymes, DNA, with this enzyme producing DNA, methylated undergoes to methylated, what happen? The methylation, due to the methylation, there is no formation RNA, there is no formation of protein, there is no enzymes. Without enzymes, I can digest the food material. That's why the delay, that might the digestion process delay could be delayed. So these are the examples. And lifestyle factors, for example, normal functioning cell without methylation, the perform the cell well, but when you smoke or any untoward activities, automatically if the cell undergoes to methylation takes place, there is no malformation takes place. It leads to cancer also. So they are all the methylation process. And installation, another one in installation. In installation, what happen? In installation means remove positive charges, thereby reducing the affinity between the histone and DNA. Actually, the histone and DNA, they are negative and positive. That's why they are closely, the firmly attached each other. But automatically, if remove the positive charge, That means histone charges as result enhance the transcription. That means, for example, here what happened? Actually, this is the histone protein. Why the histone firmly attached to DNA? Because of the affinity between the positive charge of the histone and negative charge of the DNA. But this acetylation, what happened? This negative charge removed. When the negative charge removed, automatically the histones also removed. When the histones are removed, automatically the DNA spread like a linear. No, there is no barrier. Now, if the RNA polymerase bind, it is move freely forward direction and form the RNA strand. So this is about DNA acylation, right? And as a result, enhance the transcription. While the histone deacetylation, what happened? Repress the transcription. Acetylation enhance the transcription. Deacetylation repress the transcription. Example, MECP2 protein. MECP2. MECP2 protein, which binds to the gene. When it is bind to the gene, automatically deacetylation takes place. When deacetylation takes place, there is no remove of positive charge, there is no remove of histones. That's why no transcription and translation. This is about uh, first one, chromatin. And the next one, epigenetic control. Epigenetic, epigenetic control of gene expression. Epi means on the gene, which means not inside. The chromatin which are inside changes, but not inside the no changes in nucleotide composition. If the nucleotide changes as it is, but DNA made up of nucleotides. Nucleotides are as it is, but changes and upon means so far we discussed about methylation. DNA methylation, there is no changes in cytosine between adenine and thymine, but only added by methyl group to cytosine. And acetylation also same. If acetylation takes place, automatically the removal of protein, histone protein. That's it. There are no changes in internal nucleotides. The methylation of phosphorylation, also referred as ubiquination, is the process of uh, attracting. So sorry. Methylation phosphorylation is also referred as ubiquitination. In this process, attacking ubiquitin, small protein. Ubiquitin is the protein. This protein found in almost all tissues in eukaryote, which affect the cellular process by regulating the degrade of protein and smogulation of histone protein is a post-translational modification involved various cellular processes such as nuclear cytosine transport, transcriptional regulation, in which small protein SUMO, the name is SUMO, is covalently bound to the target protein by specific enzyme. This mechanism discovered by Fruch-McClure. Next, third one, control of eukaryotic transcription factors. What are the transcription factors? For example, if you take the transcription enzyme first, RNA polymerase 1, RNA polymerase 2, RNA polymerase 3, you know already, one for rRNA, two for mRNA, three for tRNA. So basic structure in any manner, for example, if you take the RNA polymerase 2, which synthesize mRNA from the DNA strand, in this exons also present, introns also present. and promoters also present region, regulator also present. Mainly promoter consequential sequences. A Pribno box, example Pribno box. So what happened? For example, in promoter site, if 2D not attached, not attached to promoter site, it is inhibited. For example, it is inhibited 2D, cannot recruit the A, cannot recruit the E and G and F, cannot bring the polymerase enzyme to promoter site. Without polymerase enzyme, how can prepare the mRNA stand? That's why this is controlled by the factors. This is one of the example for two. And any other examples here given? Regulation eukaryotic, switched on, switched off, okay. Chromatin structure, epigenetic control completed, transcriptional initiation completed, transcript processing and modification. Already told you transcription processing and modification. That is 5-methyl cap. I again write the same 5-methyl cap formation, polyadenylene formation, and spliceosome splicing formation. RNA transport, transcriptability, translation initiation, small RNAs control transcript level, post translational modification, protein transport, control protein stability. Actually, you are given 11. But in types of eukaryotic regulation, you can just write any two only, either chromatin structure, epigenetic control, or transcriptional processing modification, transcriptional initiation. Already so far 4 completed. But you can write any two in point of examination. And so another important one is uh, in control of eukaryotic transcription factors. Another important control, tissue specific gene expression. In this tissue specific gene expression means how is gene A turned out in the cell type, and another dependent primarily on whether the transcription factor for the gene enhancer active or not in cell. For example, this is the one cell. This is the another cell. If you take first cell, different genes, one is regulated by A, one is regulated by A, which is recognized by the transcription factor A, and the other gene is regulated by enhancer B. Excuse me. Another one is regulated by B. This is type 1 cell, this is type 2 cell. They are also same A gene, B gene. In one cell type, transcription factor A might be active. This one might be active. Of course, B gene is also present. Okay, what you say? B gene also present but not active. And right, might be active, whereas transcription B might not. In such cells, only the first gene would be transcribed. Only this first gene only transcribed and synthesized. Synthesize. Let's change color. mRNA. This is not active and not synthesize the RNA, not produce the protein. In other cell type, the transcription factor B would be active. This active, but A not. Only the B gene can synthesize RNA, but not A. To determine which transcription factor are active in which cell type, there are a number of mechanisms by which transcription factor can be regulated. For example, that means what happened here? So of course, if you take any cell, all genes are present, but some genes are active and some genes are inactive. For example, in an organism, so a special type of cells, for example, that give an example, heat shock genes. Actually, these yeast shock genes only for heat resistance. These genes active in cold weather? No. So the transcription factors can be activated by environmental signals. For example, usually all organisms have a set of genes called heat shock genes that encoded protein that help the organism survive the heat stress. These genes are activated under the condition of heat stress under the control of transcription factor called heat shock transcription factors. This factor is always present but is only activated when greatly increased temperature are detected. When temperature increased, automatically the gene also activate. When the gene activate, with this gene prepare the RNA, it form the protein. So this is about a specific tissue specific gene interaction. And transcription function modification. And another important one is post translational modification. Like a post transcriptional changes, post translation changes also there. That is glycolation, acetylation, alkylation. What is glycolation? Glycolation changes mean after formation protein of any way, this is mRNA strand formed for this mRNA. From this mRNA, protein formed. After formation protein, this protein attached by carbohydrates, glucose strand. This is one of the modification. That is glycosylation. Glycosylation. So many protein, particularly eukaryotic cell, are modified by addition of carbohydrate. If addition by carbohydrate, that is of course it is the protein, but it's protein added by carbohydrate. The glycosylation in protein result in addition of glycosol group, either aspirin, hydroxylin, serine, some other any type of amino acids. Next, acetylation. Simple acetylation name itself given. Acetyl group usually attached to the N terminal of protein. Alkylation, that means alkyl group, acetylation, acetyl group, alkylation, alkyl group means maybe methyl or ethyl group. Methyl or ethyl group can be added to the strand, protein strand. So biotinylation, acylation conserved in lysine residue within biotin appendages. Glutamylation, covalent linkage glutamic residue to tubulin, some other protein. Glycylation, covalent linkage to one or more fatty glycine. Glycine residue, glycylation, glycine residues to the tubulin C terminal. Isopropylene, lipolation, phosphopento phosphorylation, sulfation, they are the changes. C-terminal modification. These are the changes in translation process. So this is about eukaryotic gene expression. Right. Let's see the restriction endonucleases. Restriction endonucleases. Right. Restriction endonucleases. What is restriction endonuclease? Why did it continue? restriction endonucleases or restriction endonuclease. First of all, what is endonucleases? Endonucleases, the function cut DNA. Endonucleases cut the DNA strand. For example, this is the DNA. Cut the DNA into two pieces. This is endonucleases. That is function of endonucleases. Now, what is restriction? The restriction means the recognize the particular region. Recognize the particular region only, the particular regions only of DNA, particular sequence. The particular sequence called as palindromic, palindromic sequence. In palindromic sequence region, they cut the DNA. So that is the activity of restriction endonuclease. This restriction endonuclease mainly produced by bacteria. From bacteria, these restriction endonucleases enzymes are produced. Different type of restriction enzyme nucleases produced from the bacteria. And also green algae chlorella, from chlorella also, chlorella green algae also, but mostly bacteria. How it is produced? Actually, what happened, especially in bacteria, this is the bacteria cell. This bacteria cell, when it is infected by phase DNA, that is virus, phase virus. So phase DNA enter into the bacteria cell. When phase DNA entered the bacteria cell, this bacteria chromosome automatically activated and forms the enzymes, restriction enzymes. These restriction enzymes kills, breaks the DNA. So now these restriction enzymes separate. What happens if restriction enzyme not present, not produced? If not produced, this phase DNA integrated with the bacterial chromosome and disintegrate this bacterial chromosome. Right. So this is the production of restriction endonucleases. And for example, you might have a doubt if restriction endonuclease function is cut the DNA. Already restriction enzyme present inside the bacteria. Why can't cut the bacterial DNA? Why can't cut the bacterial chromosome? Because in bacteria, some enzymes that modify the regulation site of the gene. This regulation site genes automatically modified. So when it is modified, the restriction enzyme can't identify that region. That's why the same restriction enzyme can't cut the same chromosome. Only the restriction enzymes of bacteria can cut other organism bacteria, other organism genome. Now let's see the types of restriction endonucleases. So restriction endonuclease 1, restriction endonuclease 2, restriction endonuclease 3. Three types, 1 and 2 and 3. First, let's see the restriction endonuclease 1. Restriction endonuclease 1, the first identified enzyme, that's why it's the given name 1. This is the second, this is third. From the E. coli, from the E. coli bacteria. Coli bacteria. This enzyme cut the site the difference and in some distance 1,000 base pair away from the recognition site. For example, uh, this is the gene, this is recognition site, recognized site. From recognized site, 1,000 base pair away, cuts here. 1,000 base pairs away, cuts here. So that is recognized site is random, randomly formed recognized site, and it is composed of two main portions, 3 to 4 nucleotides, another containing 4 to 5 nucleotides. That means randomly formed. The randomly formed means this is one recognized site, another here, another recognized site. So for everywhere, 3 to 4 nucleotides. Here, 3 to 4 nucleotides. Here also 3 to 4 nucleotides are there. And here another important one is this restriction enzymes choose the bifunction. The bifunction is if the function endonuclease activity, endonuclease activity, endonuclease activity, endonuclease activity. Another function is methylase activity, methylase, methylase activity, methylase activity, both functions. And endonuclease activity, it requires ATP energy. Endonuclease activity means cut the DNA. To cut the DNA, it requires ATP energy. It requires cut the activity energy. And type 1 restriction enzyme passes three subunits. The three subunits. The three subunits are HSDR, HS, HSDR, HSDM, sorry, small, HSDM, HSDS. Another one is S. HSDR required further restriction to recognize the restriction site or for restriction or is required. HSDM is necessary for adding methyl group host. This methylation, HSDM. The name itself M for methylation. Sorry, here M for methylation. And HSDS is important for specific site, cut the site recognition. Recognition by R, the cut site by S, in addition to its methyl transferase activity. And another function is S, methylase transferase activity also done. So this is the type 1 about type 1. And second one, type 2, the restriction enzyme differ from type from enzyme in several ways. So for example, first let's see the differences. In restriction enzyme 2, unifunctional. First of all, this is bifunctional here, ATP, endonuclease activity and methylase activity. But here only unifunctional, that is the endonuclease activity, only endonuclease. So unifunctional, that is the first difference. Endonuclease activity. For methylase, another enzyme there. For methylase enzyme, another enzyme there. So methylase activity, we separate enzyme involved. And here two identical subunit. Here the three different here, but here the two the same subunit, two identical subunit, two identical, two identical subunit. And these subunits cut the restriction site only, but not far away. But what happened here? Somewhere here the restriction site, but cut after the 1000 base pair. But in this restriction enzyme here and cut at the restriction site only. That is different. And for this endonuclease activity, does not utilize the ATP. Don't use ATP. Don't use ATP. Here ATP utilized, but here the ATP not utilized to cut the genome endonuclease activity. But instead of that, it utilized some of the magnesium 2 plus magnesium factors. Utilize the magnesium factors only. And next one, 3, type 3. And type 3 enzymes to separate non-palindromic sequence that inversal oriented, I will explain. They cut the DNA about, here also same. This is recognition site. For the cut the DNA 20 to 30 base pair after recognition. Here, cut here. Here 1000, but here 20 to 30 base pairs. And for this activity, cut nuclease activity, sorry, endonuclease activity, it utilizes ATP energy. Utilize ATP energy to utilize ATP energy. And here like a restriction endonuclease 1, it is also bifunctional. Endonuclease activity and same methylase activity. Endonuclease activity and another one is methylase activity. So the functionally the both are same, but this is the unique only endonuclease activity. Two is only endonuclease activity, but one is one endonuclease and methylase. There are three also endonuclease and methylase, and one also utilizes ATP energy to nuclease activity, and the three also utilize activity ATP energy. But here no ATP energy required for the nuclease activity, right? So this is the RNA restriction endonuclease 3. And here the given the table, the nomenclature given here. Inside the table, shortcut name, enzyme names, source, recognition sequence, and cut. But here the recognition site, the clearly given the table. Don't read all this. Just this is for example. E. coli, Escherichia coli. So recognize site, cut the site. And if you see the table somewhere, now when there the given the star mark. Star marks means which gives the blunt end instead of sticky, which gives blunt end. I will explain clearly what are the blunt ends, what are the sticky ends, and what is the palindromic sequences. Right. So nomenclature of the enzyme nation, the abbreviation ECO in cable clearly given. Abbreviation E for Escherichia. Bacteria name, bacteria abbreviation. This is shortcut. CO means coli. R means type. RY13. I mean first identified. So first identified description, first Escherichia, that is the genus name, coli, species name, and R113, that is the strain, and first the order identified in the bacterium, first time, that is given the number first. So this is the identity nomenclature. Nomenclature not an important one. And recognition site. Already told you, if any in any enzymes, there is a recognition site. This recognition site that is called as a palindromic sequence. Palindromic sequence means if you read any direction, same nucleotide appear. For example, here this is DNA double strand, DNA double strand. The nucleotide sequences are, let's change color. G T A T A C. Same sequence G T A T A C. So this is the palindromic sequence. That means if you read any direction, this direction G T A T A C. The same right this direction G T A T A C. Right. This is palindromic sequence. Right. And in this sequence present in 1 and 2 and 3 also. Now the how the cut ends and blunt ends are formed. Let's see the cut ends and blunt ends. The cut ends forms cut end, sorry, sticky ends and blunt ends. Sticky ends with help of the isosegomers. Isosegomers means the restriction enzyme two types, isosegomers and neosegomers. Isosegomer restriction enzyme which cuts the uh, recognition site at the same specific nucleotide. The same specific nucleotide, for example, the same the repeated sequence. That means this enzyme cut. Let's change color. cut between GT, guanine, thymine, cut here, and same cut between GT, same sequence, cut at same sequence. So this is the isosegmers. This type of polymerases called as isosegmers. Due to this, it is forms the sticky ends. Sticky ends like G, T, A, T, A, C ends. Another one. So simple. It is separated. G. This is only empty. Here, this is actually only G present. This is to dryer. G, here C, T, T, A present. But already G cut like this. Now this is called sticky end. Now it is placing two complementary genes. When the same complementary present, now it is easily attached to there. Right. So this is called sticky end. And now neosesomers. The neosesomers, these are actually the that recognize the same sequence same, but these often cleave the different location, but not same location of the sequence. However, the different enzyme recognize and cleave. For example, the same sequence GTAA, let's draw here. G T A T A C G T A T A C. Right. Same sequence, but it is cut at different site. For example, sorry, it is not this one. CG GT like this. It is cut like this to equal parts are formed. This is called blunt end. Only GTA C A T formed one side, blunt, and TAC ATG. This is called blunt. It is not attached to another nucleotides. So these are called blunt ends. So the first one sticky ends, second blunt ends. So this is about restriction endonuclease. This is very important for short essay. If it is separated one shot, two one shot, and three one shot, right? And next one, type restriction endonucleases enzyme application. We will continue next class.